Hello everyone, welcome to today's webinar. It's being hosted by Dicel Arbor Biosciences. This session will be recorded and made available for later viewing, so we welcome all our visitors from the future as well. I'm Allison DeVault, the Director of Genomics at Arbor. I'll be moderating today's webinar. Our topic is advances in resistance gene sequencing, or RenSeq, for crops via targeted NGS. This is a two-part series. The second part will be held next month in March. This workshop was actually intended to be held at the PAG conference this past January. Unfortunately, that was canceled at the last minute due to Omicron, but we're very grateful that all our speakers were willing and able to convert to this webinar format instead. So before I get into the science, I will just point out that we'll accept audience questions for our speakers today via the questions portal in the webinar control panel. You can submit a question at any time throughout the session. We'll go ahead and curate those and provide them to our speakers to answer. Please ask away at any time. Apologies in advance if we don't have time to get to yours in the live session. One of the most agriculturally important groups of plant genes are called the R genes. This is a complex family of genes which typically encode nibbler or NLR proteins. And in plants, they function in disease resistance to many pathogens, including fungi, bacteria, viruses, and more, which makes them incredibly relevant to understand and develop in an agricultural context. RenSeq is the technique of resistance gene enrichment sequencing, which utilizes targeted next-generation sequencing via hybridization capture to enrich both known and unknown and novel R gene sequences from plant DNA or RNA NGS libraries, which are then sequenced on short or long read sequencing platforms. The enriched R gene sequences can then be digitally reconstructed with bioinformatic analysis. This is a powerful approach that can be deployed in various programs for discovery, trait improvement, and more. At Dicel Arbor Biosciences, our myBaits product line provides custom and pre-designed hybridization capture kits that can be used in RenSeq enrichment workflows. And in our myReads full-service sequencing and bioinformatics lab, our team of experts can perform any and all steps of the full NGS workflow, including for RenSeq projects. There are many publications on a wide variety of plant and pathogen species, such as wheat, potato, Arabidopsis, and more, that have used Arbor's products and services for RenSeq. I'm highlighting just a few of those there, but quite a few more are linked on our website if you're interested, where you can also find additional info about RenSeq, as well as our contact info if you'd like to reach out to us to discuss your next project with our team. For today's webinar, we're featuring talks by two researchers that are working in cutting-edge plant genomics. Our first speaker will be Robert Heal. He is a Ph.D. student in the Jonathan Jones group at the Sainsbury laboratory in the UK and is currently studying genetic resistance to two pathogens in potato, with the goal of improving disease resistance via integration or genetic modification. Second speaker will be Timothy Hewitt, who is a postdoc at CSIRO in Canberra, Australia. Tim is currently studying serial rust pathogen genomics and effector discovery. Prior to his current position, he did a PhD with CSIRO and the University of Sydney's Plant Breeding Institute, researching disease resistance genetics in cereals. So today, Robert will be presenting first on his work on the genetics of P. infestans resistance in nightshade, which is a potato relative, using RenSeq and effectoromics. So before I hand things over to Robert, just a reminder, if you'd like to ask a question throughout the talk, go ahead and pop those into the questions panel of the webinar control panel. And with that, just give me a moment to transfer things over to Robert to give his presentation. Thank you.
Okay, great. Can you see my screen?
Yes, we can. Thank you, Robert.
You can hear me. Perfect. Okay, great. So yes, thank you for the introduction. Just to repeat slightly what you said, I'm a third-year PhD student in Jonathan Jones's group at the Sainsbury laboratory based in Norwich, attached to the University of East Anglia. And I'm going to present some work which has been performed by myself and many other people in our lab and various other labs around the world towards dissecting components of non-host resistance to Phytophthora infestans in the wild species Solano Americanum. So I'll just start by briefly introducing the pathogen to you. So we mainly work with Phytophthora infestans, and this is perhaps the most famous plant pathogen which anybody works on. It is a classic example of the severity which can be caused by plant pathogens and the effect it can have on both social and economic contexts. So you can see from these three pictures the rapid effect it can have on a potato field. So in this first picture, you can see what appears to be a perfectly healthy potato crop. And then within 1 week, you can see that the pathogen spreads and you cause rapid loss of leaves across the whole canopy. And then within another week, you can see the crop is completely decimated and there's no longer any foliar canopy. And in fact, this is actually even worse than it appears because the pathogen also spreads to the tubers, causing a complete yield loss. And there's obviously a severe economic loss due to this pathogen, which is estimated between $7 and $10 billion each year. And of course, the social consequence was highlighted in the Irish potato famine in the 1840s, which resulted in the displacement and death of millions. So we in our group work towards improving potato varieties and preventing yield losses and reducing the environmental impact of fungicide sprays. So we mainly do this by transferring resistance genes, or R genes, from resistant wild species into susceptible cultivated potato. So in this case, we have our triple stack of R genes, which we've deployed in trials over the last few years. And these are Rpi-amr1, which is resistant to Phytophthora infestans from Venturi 1, and Rpi-amr1 and 3, which is resistance to Phytophthora infestans from americanum 1 and 3. And you can see these are just examples of the wild species we're using, which are from a variety of different sources. And when these three genes are stacked together, you can see the effect in this picture on the right. So there's 2 rows of potatoes here from our field trial in Norwich. You can see on the right there's the Wildtype Maris piper variety, which is a popular variety in the UK. And there's been a complete loss of all the foliar tissue. And this is in stark contrast to the transgenic line where we have our 3 R genes. And you can see there's absolutely no symptom of the pathogen at all. But obviously we want to further improve this and make it more durable by stacking additional R genes. But to do this, we need to first clone them. So these 3 genes were cloned over a period of around 15 to 20 years. And with resistant gene enrichment sequencing, we can now rapidly clone more. So using RenSeq, which was originally developed in the Jones group, we can enrich for resistance gene encoding DNA and then sequence them and characterize the allelic variation between different accessions. So in our case, we use our version 4 of our Solanum bait library, which was developed by Burkhardt and Camille from the John Innes Centre and the Sainsbury laboratory, respectively. And this is based on 22 different genomes of wild Solanum, and it includes 60,000 RNA baits. And using these biotinylated RNA baits, we can mix them with gDNA from different samples, so in this case from a bulk susceptible group and a bulk resistant group, and then we can enrich using that biotin tag to have an enriched sample which is containing our NLR encoding genes. And then we can combine this with various sequencing platforms, whether they're short read or long read, and then assemble based on that. And this figure on the right just highlights the use of this. So this is showing the different, showing all the NLR encoding genes from Solano Americana, which are identified using RenSeq. And you can see many of these gene families are very complex and have around 20 different paralogs. And using these long reads in combination with RenSeq, we can completely resolve them and identify all the NLR encoding genes in a certain accession. And we now have a fairly streamlined pipeline for identifying our genes. And the first step of which is to perform a long read RenSeq using PacBio and the myBaits. And then we can combine this with a short read Illumina RenSeq based on different bulks from bulk resistant, bulk susceptible, identified from a segregating population. And then we can identify resistance linked NLR encoding genes from our resistant line. And then using techniques such as cDNA RenSeq, where we enrich resistance gene encoding genes or transcripts. We can identify which of these candidates are expressed, and then using those transcripts, we can identify the gene models and find whether they contain all of our canonical domains which we'd expect from a resistance gene. And then it's just the simple matter of testing to find which one is the functional paralog. But to do this, we have our favorite source of resistance gene, which is Solano Americana. And this is a wild diploid species which has a worldwide distribution and is very amenable to genetics due to its ability to be inbred and also have complete self-compatibility. And you can see that there's an example of a detached leaf assay on these different americanum accessions on the right. And this is just for infection with Phytophthora infestans. And you can see in the accession at the top, there's complete susceptibility, and it's spread across the whole leaf. And many of the other accessions show complete resistance. So either this small pinpoint hypersensitive response or just a completely green leaf. And we have our 54 accessions at TSL, and we can use these assays to identify which of them contain resistance. And this is just indicated in this table on the left. So all these green bars indicate accessions which have complete resistance, and the orange or red have some susceptibility in these leaf assays. But the important thing to note is that whilst we have susceptibility in detached leaf assays, in fact, all of these accessions are completely resistant in the field. So this is just a point of why we call americanum a non-host species to Phytophthora infestans. And we've built up quite a variety of genetic resources for americanum. So we've performed RenSeq on 18 different accessions, and these are just selected based on interesting resistance phenotypes or our lab susceptible at the top, which is 2-7-1. And we actually use this data not just to clone resistance genes, but also in collaboration with others. So we're currently collaborating with S.am, whose lab is based in Germany, and he's studying recombination between NLRs as a route to genetic diversity. And he's using our 18 RenSeq data sets to do this. And we're also, in addition to using RenSeq, we have developed 4 reference genome quality assemblies based with 2 different collaborations. So we collaborated with Ki-Hoo and Sohn's group from Korea to sequence the SB2273 and 2275, and with Simon Wang's group from based in China to sequence 2271 and 1102. So with our combination of our RenSeq data and our whole genome data, we can now rapidly position genes within the genome and identify co-segregating candidates. But to identify new resistance genes, we first have to think about what we've already done. So previously, postdoc Camille Vutek has cloned 2 resistance genes, which are Rpi-amr1 and Rpi-amr3. These were cloned from accessions 2273 and 1102. And you can see the effect of these genes in these transgenic potato lines. So on the left, each picture is the transgenic line carrying the functional paralog. So this is Rpi-amr1e and Rpi-amr3i. And on the right, you can see the non-functional control, so the 3A paralog and the 1A paralog. And you can see the functional paralogs confer complete resistance, and there's no sign of infection at all, which is quite a contrast to the non-functional genes. And more recently, postdoc Xiaolin has identified the 2 different recognized effectors of these genes, which are AVR-amr1 and AVR-amr3. And you can see that by co-expressing these with the corresponding resistance gene, you can see a strong HR in Nicotiana benthamiana. And we can apply these and simply transiently deliver them via agrobacterium into our americanum collection. And then we can screen to see which of our 50 accessions have Rpi-amr1 and 3. So this is these 2 additional columns just represent that. So where there's a red bar, we see a strong HR response when these effectors are delivered into americanum. And where there's a green bar, we see absolutely no response. So you can see that for our susceptible lines, none of them contain Rpi-amr3 or 1, as indicated with these rows. And you can see that most accessions actually contain both resistance genes, or at least 1. But there are 2 other accessions which contain neither and show strong resistance, and these are 2275 and 3370 down here. And so resistance in SP2275 is what we call Rpi-amr5. As I said, this contains no functional Rpi-amr1 or 3, so we know it's a novel source of resistance. And you can see that it shows a fairly strong resistance phenotype compared to our susceptible line. And when we simply make a segregating F2 population, we can see that resistance segregates in a 3-to-1 ratio, indicating a single gene is responsible. So we've performed our mapping pipeline on this accession, and we can see that by using bulk segregant RenSeq, we find a cluster of 16 NLRs where Rpi-amr5 matches to. And with the other accession, SP3370, which we call Rpi-amr12, there's again no functional Rpi-amr1 or 3, and we perform the same pipeline with bulk segregant RenSeq and Bioinformatic. We find that Rpi-amr12 matched to the same cluster as Rpi-amr5, and indeed the Bioinformatic assemblies show absolute identical haplotypes. So these two genes are in essence identical. So to identify the AMR5 paralog conferring resistance, we have to look deeper into this cluster of 16 NLRs. And this is just to represent those different NLRs within the cluster. So I have 16 different paralogs to look at. And using cDNA RenSeq and looking at the gene models and identifying canonical NLR domains, we can find that most of these genes can be excluded. So many of them I've indicated as partial NLRs, meaning that they're lacking either the CC domain, the EMBARK domain, or the LRR domains. And an additional 3 genes show no expression in cDNA RenSeq data. And this leaves us with 4 good candidates for Rpi-amr5. So normally when we look to test resistance genes, we can use Nicotiana benthamiana as a model for this. So in this picture here, we've expressed Rpi-amr3 and then drop inoculated with P. infestans. And you can see after a week, there's absolutely no pathogen growth. And we know that Rpi-amr3 works very well in this assay. So you can see there's only these very small indications of where the inoculation was. So we can use this assay to test our 4 candidate genes. However, we see a completely different result for most of them. So for NLR3 and NLR6, we see no function in conferring resistance in these assays. In fact, we see pathogen growth spreading from the inoculation points. And for NLR9 and NLR13, we see this wilted phenotype across the whole leaf. And this is not due to infection. This is in fact due to autoactivity of the NLR in Nicotiana. So this left us with a slightly difficult situation of how to test the genes. And we thought that rather than having an increased resistance assay, we can in fact use virus-induced gene silencing to test for loss of resistance in the americanum accession. So this is a relatively simple assay to perform. And just looking at the our expected transcript of an NLR, we have the CC domain, the EMBARK domain, and the LRR domain. And we can silence the entire cluster by targeting the EMBARK domain. So EMBARKs are generally conserved within, or highly conserved within clusters. And in fact, we can use a nearly, 265 base pair fragments in this case, which has greater than 90% identity between those 16 paralogs. And then we can target individual paralogs more specifically using the BIGS constructs targeting the LRRs. So in this case, we have constructs which target 2 different regions within the LRRs, each with a small 60 base pair fragment. And this is where the paralogs are most diverged. So applying this to our candidates, you can see from the negative control of NLR16, so this is one of the genes we know doesn't work. You can see that upon silencing and inoculation, there's no growth of the pathogen from any of these inoculation points. But when we silence the entire cluster, you can see widespread susceptibility. So this confirms the identity of AMR5 as a gene within this 16 paralog cluster. And then targeting those 2 autoactive genes separately, you can see by silencing NLR9, we see no loss of resistance, but by targeting NLR13, we see pathogen growth from all these droplets. So this means that at least for now, we are fairly convinced that NLR13 is the functional Rpi-amr5, and we are working to confirm this in various different ways. So we're generating stable transformants of Solano Americanum, expressing NLR13 under various different promoters. And we're also working to knock out NLR13 in our resistant line and test for a loss of resistance. So I'm going to move on to a slightly different part of our project now. So interestingly, as I said before, that americanum, whilst we see some susceptibility in detached leaf assays, we in fact see complete resistance in the field. So this is a picture from a couple of years ago, just after it's just after the pandemic started in the UK, and we managed to sneak a few americanum plants into the field. And you can see that this is just right next to our GM potato field, which is in the background here. We have plenty of plants in this GM plot, which we use as susceptible controls, and showed strong P. infestans infection. And just by planting our americanum nearby, we hope to catch some infection on many of them. But in fact, we didn't see any infection on any of these susceptible lines, indicating they all have fairly strong field resistance. So we're questioning what the basis of this field resistance is, and we hypothesized that it could just be simply conferred by NLRs. So to test this, we first thought about looking into whether these americanum accessions have recognition of P. infestans. And to do this, we use effectoromics. So we screen a collection of effectors from P. infestans and test for a response to any of them. And to do this, we have a library of around 500 RXLR effectors, and the RXLR domain is associated with secretion into the host from P. infestans. So using this library and work mostly led by Dr. Shaolin, he's produced this wonderful figure here. So you can see in this table, there's basically for every row is an americanum accession, and for every column is a different P. infestans effector. So you can see there's three different phenotypes we have from this. We have the lightest color, which is showing no response, the red, which indicates a strong response, and then an intermediate response, which is this sort of color in between. And you can see that all americanum accessions have recognition of at least, far more than one or two effectors. So some of these are recognized by all accessions, which could either be due to recognition or due to toxicity of the effectors. But most effectors have a differential response between our different accessions. So this is good news because it confirms that the field resistance could be due to NLR recognition, and and all americanum accessions have recognition of P. infestans. So based on our theory of multiple recognitions conferring field resistance, we're working to map and clone several recognition genes. And the first I'll talk to you about is recognition of this effector, which we call effector 344. And this is a gene which maps to a chromosome 1. And you can see from this table on the left, there's a differential response of around 50-50% of accessions recognizing or not recognizing the effector. And just to highlight, our favorite susceptible plant, 2271, has recognition of this effector. And we have low coverage Illumina sequencing of whole genomes for all of our accessions, and we can have a rough look using GWAS to identify any loci which could be candidates for this. And you can see that the greatest peak is on chromosome 1 towards the top of the chromosome. But this is fairly weak evidence because we only have around 50 accessions, so GWAS is maybe not the most reliable method to use for this. But indeed, we can just perform simple mapping in an F2 population and use bulk segregant RenSeq to identify any loci. And indeed, we can confirm that this point at the top of chromosome 1 is co-segregating with recognition of effector 344. And in this case, we were fairly lucky with this locker, so it only contains two NLRs, one TR NLR and one CC NLR. And using cDNA RenSeq on 2271, we can see that the CNL is expressed and the TR NLR is not expressed. So simply just our next step was to clone this gene and test it for recognition in transient assays, which is exactly what we did. So we cloned two different alleles, one from 1102 accession and one from accession 2271, and we transiently delivered these in benthomiana, where either individually or with effector 344 or with Rpi-amr3. And you can see that for the 2271 allele, we see some HR, which is associated with autoactivity. So when the receptor alone is expressed, it still causes a somewhat response, although the response with effector 344 is slightly stronger. But with the 1102 allele, we can see that there's no residual or background HR, and there's a strong response with effector 344. So this is good news, and we managed to very rapidly clone this gene. So it's indicative of how we hope for the next few mapping exercises to go. And the question we wondered immediately was whether overexpression of receptor 344 could confer resistance in Nicotiana benthamiana. So to do this, we transiently delivered receptor 344 into benthamiana leaves and tested with a drop inoculation of P. infestans. So with our functional Rpi-amr3 as a strong positive control, you can see that there's no infection points in this example. And with the non-functional 3A paralog, we can see that there's widespread growth from the inoculation point. And then when we have receptor 344 overexpressed in benthamiana, you can see that there is some lesion development, but this is far less than the negative control. And you can see the quantification of this on the right. So you can see we've measured lesion diameter on various leaves of expressing each of these constructs. And you can see that for receptor 344, the lesion size is smaller than the Rpi-amr3A control. So we're working towards further characterizing this, and we're wondering if by using CRISPR to mutate receptor 344, we can remove field resistance from one of our lines. And we're also adding receptor 344 into Solano tuberosum in an effort to see if we can increase resistance with this fairly partial resistance gene. And we're also looking towards cloning 7 other effector recognitions from americanum. And I'll just briefly talk about one of the others. So this is effector 16, our lab ID for it, and you can see on the left with our americanum collection, we can see that only 5 accessions recognize this effector. So we couldn't take a GWAS approach with this one. And again, it's recognized in our susceptible line 2271. And then by simply performing mapping in an F2 population using bulk segregant RenSeq, we identified a locus which contains 2 different clusters of NLRs. So each of these red annotations represents 1 NLR. And then by doing some marker screening and fine mapping, we identified a 300 kilobase interval, which contained just 1 of these clusters. And this has 7 paralogs in SP2271. And by using cDNA RenSeq, we identified the full reading frames of all these paralogs and found that all 7 were expressed. So then the next step is just to clone all these candidates and test them by co-expression with effector 16. And this is exactly what one of our predoctoral students has managed to do in the last few months. So you can see some transient expression assays in Nicotiana tabacum here. And you can see that there's 6 candidates which we cloned, which were NLR 12, 13, 14, 15, 16, and 17. And you can see when these are co-expressed with Rpi-amr3, there's no HR at all. And when one gene, NLR 16, is co-expressed with effector 16, you can see that there's a very strong HR response. And this is what we now are calling receptor 16. And the difficult thing with this gene is we can't quite repeat what we saw with receptor 344, because the one allele we have cloned is autoactive in benthamiana. So we can't transiently deliver it and inoculate and test for partial resistance in the same way. But as I mentioned, there are some other accessions which carry this recognition. So we're currently cloning the different alleles and testing for autoactivity in benthamiana. So I'll just finish by summing up what I've said. So resistance gene enrichment sequencing is a useful method for defining variation in NLR encoding genes. And that enables us to rapidly clone our genes from our americanum accessions. And whilst most of these accessions we have carry functional Rpi-amr1 and Rpi-amr3, we know that there's other strong resistance genes present. And one of these is Rpi-amr5. This gene maps to a 16 paralog cluster, and we've found that 1 paralog appears to be implicated in resistance in our VIGS experiments. And even by looking at supposedly susceptible accessions of americanum, we can find that they all have field resistance. And we're trying to dissect the components of this using effectoromics and RenSeq to rapidly clone these recognition genes. And of our 7 which we're prioritizing, we've now managed to clone 2 of them. And these genes, whilst maybe not conferring strong resistance, do confer some partial resistance in our transient assays. So I'm just going to finish by acknowledging all the people who contributed to this work. So I'll first acknowledge Jonathan Jones, who's my PI and very kind to give me a PhD position in this group, and Shaolin, who I've shown most of, well, some of his work in americanum and with his effectoromics. Then we've had several great predoctoral interns over the last few years, and also former postdocs in our group who've performed a lot of work in developing the americanum collection. And I'll thank our collaborators in Korea who've generated 2 of those reference genomes, which we'll be publishing soon. And again, 2 reference genomes were developed by our collaborators in China. And I'll also thank the support teams at TSL who make our life much easier. And if anyone has any questions which they want to ask but don't want to put in the chat, you can feel free to email me at this email address. Okay, thank you for listening.
Thanks very much, Robert. That was a great presentation. I haven't seen any questions come through the questions portal yet, so while you're all thinking about those questions, digesting what Robert just said, I have at least a couple I'd like to ask you quickly. The first one is, it seems to me that the collection of accessions for Solanum americanum seems incredibly important to this whole process, right? Because that's a source of this, our gene diversity. I was just curious to hear more about how those accessions are collected and curated. How is this collection developed in your group?
So for our side of collecting them, we well, I didn't do it, but years ago, Camille contacted seed banks from various parts of the world, and they have collections from expeditions in the 20th century and before, and they just share the seed with us. So we have knowledge of the collection location. Whether that's entirely reliable or not is maybe slightly more questionable. So, yeah.
Great. Sorry, I do see a couple questions here, so apologies I butcher some of your names. Andre LaRoche asks, can you comment on the impact of 35S promoter versus a native promoter?
Yes. So at least in transient tests, we use both. So we normally use 35S for the initial experiments, and then we'll reclone them under native and retest them that way. But some of these native promoters, the NLRs are very lowly expressed, so sometimes they don't have excellent response in transient tests, but work very well in stable transformants nonetheless.
Great. Question from Scott Monsma. Is S. americanum polyploid, and how much variety in ploidy in the Solanum is there in the Solanum group?
So americanum is a diploid, but we have Solanum nigrum, which is a hexaploid and is closely related. So and there's also other tetraploids within the same group, so Solanum villosum and Solanum scabrum. And if we look for resistance in these accessions, it's obviously a lot more complicated to work with the genetics, but we once we've identified the effectors, we can in fact look and see whether these resistance genes are found in those accessions as well. So we know that nigrum, villosum, and scabrum also contain Rpi-amr1 and 3.
Follow-up question that I have, would you be able to comment on how the effector libraries themselves were built? It sounded like there were a few hundred different components there. just curious on how that gets built and maintained.
Yeah, so for many of them, they were identified by different groups. So I think Paul Birchley's group in James Hutton and Vivian Bluesand's group in Wageningen, and from Sophie and Commun's group in TSL. And we've sort of made a collection based on theirs, and also identified several other ones. So with PenSeq from a paper a few years ago, it was, we identified a collection of effectors from various different Phytophthora infestans isolates and built those into that collection as well.
And a question about the sequencing technologies. It sounded, I think, that a lot of the long-range sequencing was PacBio. Have you used nanopore yet for any of that work in your group?
For enrichment, we've not used nanopore and enriched with gDNA, but we have used it with some cDNA and successfully enriched with the baits. I always prefer PacBio though.
Great, thanks. I'll just do one more here, and then if there are more, we can put those on at the very end after Tim's presentation. So last question here from David Botero. Thank you. Is there some statistical threshold in the number of vegetable materials that you should use in RenSeq?
For doing the bulk sequencing, I imagine that's the question. Well, we normally just go for 30 resistant accessions in a bulk and 30 susceptible. I think it works relatively well if you have more than 20, but the more the better.
All right, well, as I said, if there's any more questions that you guys have for Robert, please do keep putting those in the questions, and we'll try to get to those at the end. If not, we'll make sure he receives them in email. So next up, we have Tim Hewitt from CSIRO in Canberra, Australia, who will be speaking today about his work on cloning R genes in bread wheat that combines mutagenesis and RenSeq. So yeah, once again, please use that questions panel if you've got any questions for Tim, and just give me a sec and I will transfer control over to him. You see your screen?
Hello, everyone. Can you see my screen?
Thank you.
Okay. Sorry, just one moment. So, yeah, hi, everyone. So today, I'll be discussing some work that I was involved in during my PhD in cloning R genes using RenSeq. So I'll sort of more be focused on the technique today. So I won't be showing a lot of results, but I'll be discussing mostly the workflow of RenSeq and validating a candidate gene once you have found a candidate gene using this cloning technique. Yeah, I don't need to stress the importance of wheat as a crop, and it's highly susceptible to a range of fungal pathogens. So the ones I've mostly worked on are powdery mildew, leaf rust, and stem rust, which can cause many losses in yield globally. And these are biotrophic fungi. So it is important to ascertain genetic resistance against these diseases. So in wheat, there are two classes of disease resistance genes. There are the APR genes, which have a moderate effect, broad spectrum, and operate on using non-canonical mechanisms. So they're not necessarily part of the plant immune system. Whereas R genes produce a specific hypersensitive response. They show specificity to pathogen races, and they are part of effector-triggered immunity. So you can see that here. This is a resistant response showing some chlorosis around sites of infection to limit the spread of disease. So R genes usually encode nucleotide binding leucine-rich receptors, or NLRs. In wheat, they are usually a single dominant locus. They have high intraspecific diversity and they evolve rapidly. So it's very important to clone R genes to generate perfect molecular markers for marker-assisted selection and resistance breeding, and also discover novel R genes which can be used in agriculture. And also the ability to create GM stacks to make transgenic wheat that's resistant to most pathogen diseases. So one example of this was published last year where members of our lab created this Big 5 wheat, which contains 4 R genes and 1 APR gene in a single gene cassette that had strong resistance against all the pathogen, stem rust pathogens that were tested against. So technology like this can increase durability and increase the spectrum of races that it is effective against. So with traditional cloning, because the wheat genome is so large and polyploid, I mean, it's up to 17 billion base pairs and it's mostly repetitive non-coding elements. This makes like map-based cloning very laborious and slow, and also relies on genetic recombination, which isn't always feasible. So with modern cloning techniques, using NGS have helped to rapidly speed up this process. So in the wheat genome, there are around 3,400 NLR loci that have been annotated. Most of these are probably pseudogenized, but anyway, 200 rust resistance genes have been cataloged in wheat, but only around 30 or so have been cloned. And more than half of these have been cloned in the past 5 years, owing to advances in sequencing technology. And around 10 of these have actually been cloned using some form of RenSeq. And this is able to provide massive complexity reduction, which reduces the search space for a gene candidate. So the beauty of NLRs is that they can be easily predicted from translated DNA sequence, and this has allowed design of bait libraries using NLR sequences predicted from a range of Triticea genomes that have been sequenced and assembled to create these 120 base pair RNA baits. And the complementarity of this isolated DNA to these baits allows enrichment of LNR encoding fragments. So usually a minimum of 80% identity is required for capture of a fragment. But this can result in 500 to 1,000-fold increase in NLRs genes enriched relative to other genes. So in a large genome such as wheat, for example, whole genome shotgun or sequencing is not going to have enough coverage to reliably sequence all these NLRs. So enrichment is very important to capture the whole NLR set. So with the bait libraries, for Triticea, there are currently 3 available, version 1, 2, and 3. So version 1 incorporates sequences from common wheat, as well as some diploid ancestors, and also barley, and that consists of 60,000 baits. So version 2 had some addition of dicocoides and additional varieties of bread wheat, and this had improved sensitivity. Whereas version 3 incorporates a lot more varieties. So this is more improved in sensitivity, and it also includes some conserved intron sequences, which is quite useful, as well as Rye. And these can be found, so the sequences for these can be found on this website here. So RenSeq, or mutagenesis and R gene enrichment sequencing, is basically RenSeq performed on chemically induced loss of resistance mutants. So because these target R genes are a single dominant locus, they can be easily isolated if the same NLR is mutated in all or most mutants. So for example, if you have a library of NLRs, there's going to be mutations scattered across all of them, across your various mutants. But ideally, you'd want to find an NLR where you have an independent, unique mutation in each mutant coinciding at that particular NLR, and then that gives you a pretty good candidate that you can go on and validate. So some of the work I was involved in was cloning these genes, SR26, which is a translocation from tall wheatgrass, PM1, which is originated from an unknown introgression or rearrangement. SR27 was transferred from rye to triticale, and LR13, which is from wheat, but it's in a centromeric region. And all this low recombining alien chromatin, or basically it doesn't recombine in common wheat, so this prohibits map-based cloning. So these were perfect candidates for using RenSeq. And I should note that SR26 and SR27 are effective against UG99, which is a particularly threatening race of stem rust that emerged in the 90s. That had overcome most resistance genes deployed at the time. So cloning these genes is very important for future food security. So the way chemical mutagenesis is performed is using ethyl methane sulfonate. So this usually creates transition mutations. However, interstitial deletions and transversions are sometimes observed. So seeds are treated with an EMS solution of 0.2 to 1%. So usually a kill curve analysis is performed first to find the optimal concentration that will cause 50% mortality. So usually around 0.5 to 0.6 is the standard used. So for wheat, usually 700 to 1400 M2 plants are screened with a mutation rate normally around 0.5 to 1%. So this leads to the recovery of 3 to 10 mutants. However, this can vary depending on the target gene, the EMS concentration, variety, and seed conditions. And it is important to use a pure stock or near-isogenic lines to reduce residual heterogeneity because you don't want that confounding downstream analysis. So the wild type resistant seeds are treated with EMS, and they actually advance to the M2 generation and screen with a pathogen race avirulent to the target gene. And so you're looking for loss of resistance or susceptibility. So it's important to screen the M2 generation because the M1 generation, most of the mutations will be heterozygous, and because our genes are dominant, you're not going to see that susceptibility emerge. So a round of selfing is needed to see that mutation emerge as a susceptible phenotype, and so these susceptible individuals can then be advanced for RenSeq analysis. So both the both the wild type and mutant sequences are recovered. So using the myBaits protocol, the high molecular weight DNA is extracted from leaf tissue, hybrid capture is performed, and then that enriched DNA is recovered and deep sequenced. So once you get your sequencing back, so in our case, we used Illumina sequencing. The wild type fragments are assembled into a wild type reference assembly to which the mutant short reads are aligned to, which we can then screen for mismatches or Snips that are likely caused by EMS mutagenesis. So that might look something like this. So this is a Genome Browser where you see the individual reads aligned to this contig here, and then this is summarized by a coverage histogram up the top. And this SNP, which is in green, so it indicates a G to A transition. It's present in all the reads, indicating that it's homozygous and it's likely an induced mutation. So in terms of the wild type assembly, it usually generates, it's still quite a lot of data in that the number of assembled sequences you get is around 200 to 300,000. So a lot of that is just rubbish, which you can discard, a lot of these sort of small contigs, because most NLRs you're looking for around in the 2 to 8 kb range. However, they can be fragmented in your assembly, so you don't want to ignore too many of these contigs. So when you identify candidate contigs, ideally you want to see something like this contig number 5 here, where you have a unique independent SNP in each of the mutants. But sort of artifacts and noise can crop up, which you want to filter out. So here, for instance, if you have a SNP shared by more than one mutant, it's likely not an EMS SNP and could just be natural variation between the mutants, because it's very unlikely that you have an independent SNP occurring at the same base, unless you've somehow accidentally included siblings in your analysis. A SNP that's shared by all mutants and the wild type is probably a assembly error. In the case of number 3, this could be just very poor assembly or misalignments, and same as number 4. So some of those artifacts might look like this. So the one on the left, for instance, it looks like there's contribution of 2 sequences here. So this could be indicating that you have reads from 2 different haplotypes aligning, or it could be that the assembly is collapsed, has collapsed to 2 paralogs or related sequences. So that is one risk in RenSeq. Here on the right, you have this highly incoherent alignments with high mismatch density. So in a lot of cases, these can actually indicate deletions, particularly if there's sort of low read coverage compared to the clean alignments. And that's because you get, depending on the aligner you use, you get secondary alignments sort of filling in the gap from related sequences. So there's 2 bioinformatic pipelines that we use. So the one by Burkhardt at John Innes Center called Mutant Hunter, that was sort of the original one. And then as part of my PhD, I wrote my own called Mutrigo. So these perform a lot of that noise filtering and candidate discovery. So ideally, this is what you want to see from your RenSeq analysis. You want a single candidate that looks like this. So here you have the NLR motif annotations up the top. So this is the NBARC domain on the left and the LRR domain on the right. So the wild type should be free of any mutations, and the mutants should have a unique independent SNP. So in this case, all 6 mutants have a SNP coinciding in the same NLR sequence. So what if this is just, you know, this is not really your target, but these SNPs just occurred in the same NLR by chance? Well, true SNPs should be homozygous, absent in the wild type, and unique to each mutant. So in my analysis, usually around 10% of NLRs contain a SNP, one of these true EMS SNPs. So you could say the probability of an NLR containing a SNP is 0.1. So the probability of 6 independent SNPs in the same NLR by chance is 1 in a million. So using using these 6 mutants as an example, you know, is a 1 in a million chance that, you know, the null hypothesis is that this just happened by accident, that you've just found this by accident. So you can be fairly confident that this is your target gene. So 2 or 3 mutations do arise frequently by chance. So really you want more than 4 mutants to be confident in a candidate. And of course, you still need to perform validation as well. So another issue is that the assembly can be fragmented, particularly if it's a Illumina short read assembly. And the target gene can actually be split across multiple unjoined contigs. And this arises from deficient sequence capture. So there might be low bait sensitivity. Your particular target might not be well represented in the bait library. And also there could be intervening introns, which are also not represented in the bait library, that leads to gaps, and the contigs just don't get joined. So one way to overcome this is what I call fragment fishing. So it's identifying contigs whose mutations and or motif compositions complement each other. And this is where genome references can become really helpful. So for instance, if I found a contig that had only 2 independent mutations, I could blast this to a reference sequence such as Chinese Spring and identify a homologous NLR, which I could then blast back to my RenSeq Wildtype Assembly and look for hits where the other mutants complement or have mutations which complement the mutations in my original contig. And also another thing to look for is the motif composition. So say if contig 1 only had motifs for the NBARC domain, then I'd want to make sure that the other contig that I identify has motifs for the LRR domain. And then this can be later confirmed with PCR. And the reason I bring this up is this is exactly what happened with the PM1 candidate. So here I've joined the images, but originally these 2 contigs were separated. So originally it was this contig which had 1, 2, 3, 4, 5 independent mutations. And so I had to perform a fragment fishing to find this other contig containing the complementary CC and P-loop motifs that were missing from the original contig. And luckily, the mutant that had the missing SNP in the original contig, it had that SNP in the new contig that I found. And this was confirmed with PCR, so primers that joined these 2 contigs together confirm that they belong to the same gene and that there was an intron here. So Sanger sequencing confirmed these SNPs as true. Used 5' and 3' rays to confirm the gene structure. A marker based on that candidate also co-segregated resistance in 233 recombinant inbred lines and a panel of genotypes present absent for PM1. And the flanking regulatory elements I obtained with from a chromosome assembly of the wild type that was generated using RenSeq. So you can see here that intron is what was hampering, finding a single candidate, and this gene was 964 amino acids, and all the SNP mutations were non-synonymous, could be accounted for, so they cause an amino acid change. So the final validation came with transgenic complementation. So the full gene with its endogenous promoter was transferred into Fielder, a susceptible variety. And this yielded 10 independent transformants that all gained resistance to this particular race of powdery mildew. So you can see here the resistance phenotype is quite strong. Similar to the wild type here, and these did not contain the transgene, and these did. So it's quite clear that we'd found our gene. So SR26, similar story. So this actually included a deletion mutant, which was a known deletion mutant, but it was quite useful to include this in the analysis, because it meant that we could eliminate a lot of contigs where you did have read alignments for mutant 150S. So that also helped to reduce the search space to contigs that did show evidence of deletion in this mutant. So that was, the structure was also confirmed with 5' and 3' race. The regulatory regions were obtained using genome walking. And transgenics were made using, 3 sets of transgenics were made using the endogenous promoter, the SR22 promoter, and SR33 promoter, which were previously cloned R genes. So in total, there was 57 independent transgenics, and they all gained resistance to stem rust. So for SR27, this also was fragmented across 2 different contigs, and this also included a deletion mutant, which we did not know at the time, but we could validate that later on. So yes, originally it was this contig that was flagged by the analysis, and then we used fragment fishing to find contig 2413. And bridging PCR again confirmed that the sequences did belong together. Structure was confirmed from RNA-seq transcript of the infected wild type. And 1 SNP and 3 deletion mutations were confirmed in another 4 additional mutants. So there was 8 mutants confirmed altogether. And transgenics were made using a ubiquitin promoter and cDNA to generate 3 independent transgenics, and that all gained resistance to stem rust. And this transgene also co-segregated with resistance in the T1 progeny. So for LR13, there were actually 20 independent mutants from various backgrounds. These were sort of historical mutants that had been around in our stocks for a while. So 4 were used for RenSeq using the version 1 library, and actually these 4 turned out to be deletions. So I know there is read alignment, but this high density of mismatches, this noise, actually was indicating a deletion. So these were actually secondary alignments from a related sequence. And fortunately, there was some previous mapping work done, which delimited LR13 to a sort of a 20 megabase interval, which is still quite large, but luckily only 3 NLRs were annotated in this interval on the Chinese Spring reference. So one of those NLRs had close homology to our candidates. And a marker based on this candidate was tested on a panel of recombinant inbred lines and genotypes, and this co-segregated with resistance in all of them. So this was why we chose to follow up on this candidate. So a total of 23 deletion mutants and 6 point mutants were confirmed using PCR on those other mutants that we had. So the 4 that we used in RenSeq were from egret, and PCR showed that they were deletions, but we were able to cover point mutations from these other mutants here. So gene structure was confirmed from RNA-seq transcripts on the infected wild type, and flanking regions were obtained from wheat reference assemblies of mace and cadenza, which carried LR13. So the existence of reference sequences, reference assemblies were highly useful for cloning LR13. Otherwise, it may have been missed. So from the RenSeq transcripts, we had our own RenSeq experiment, but we also found transcripts in public data because it is quite a widespread leaf rust gene. And they all confirm that it has this sort of massive 15 kb intron in the 5' UTR, which made it difficult to maybe use for transgenic complementation, because we weren't sure if that intron was important for expression or not. So so we actually cloned LR13 at the same time as another group who we were in close talks with, and we published these as companion papers. So originally they thought they had a novel leaf rust gene, which they called LRZH22, which actually, when they compared their sequence to ours, it was exactly the same. So they'd actually just found LR13. So for our validation work, we conducted virus-induced gene silencing to show that when we targeted this candidate, we could suppress resistance, whereas our companion authors from Chinese Agricultural Sciences and Hebei Agricultural University, they generated transgenics themselves just using the cDNA without that big intron, and it still showed a gain of resistance. So this was fairly strong evidence that we got our gene. So overall, like once you have a candidate from RenSeq, there's a few things you need to confirm. So is it a functional NLR? Does it contain the full domain complement? Is it a complete sequence? So you might have to perform genome walking or look for the complete sequence in a reference, or perform RNA-seq or race. Are the mutations non-synonymous? So you want to make sure that the mutations cause an amino acid change. Do you have additional mutants that can be explained and confirmed? Is it supported by comparative genomics? So if you're working with a really novel gene, then you might not have a relevant reference genome, so this wouldn't apply. If you do have a segregating population, does it co-segregate with the resistance phenotype? And finally, the clincher is, can it confer resistance in an otherwise susceptible host? So can you use transgenic complementation to really confirm that it is you found your target gene? So from this work, there are a few considerations. So yes, RenSeq is really powerful. It overcomes lengthy, laborious bottlenecks of map-based cloning. But in the end, it is a hit or miss approach. So you either find your gene or you don't. So there is always that risk. So one way to maybe mitigate that is screen your mutants for potential deletions if linked markers are available, because like LR13, you don't want to include too many deletion mutants in your RenSeq analysis, particularly if absent supporting resources such as prior mapping or a relevant reference genome. You want to increase the number of mutants you use in RenSeq to be confident that you will recover a sufficient number of independent mutations. In the cases of fragmented assembly, improved libraries may overcome this. So like I said earlier, with version 3 library, it includes conserved intron sequences. So that could help to bridge some of those gaps that we saw earlier where there was an intron that was not enriched in the data. And also, I think the use of long reads for the Wildtype assembly can also recover full-length NLRs. It's more likely to recover full-length NLRs. So I think long reads are compatible with the myBaits protocol. And another thing to consider is that your target R gene, even if it is race-specific, it may not necessarily be an NLR. So in recent years, some R genes have been cloned that are actually members of these tandem protein kinases. So, you know, in the future, is it possible to maybe integrate this into existing NLR libraries or create a new class of library for this type of R gene? You know, so that's something to think about. So I'll just wrap up by thanking my PhD supervisors, Evans Laguda from CSIRO and Peng Zhang from University of Sydney, as well as Robert McIntosh, Jianping Zhang, who was the lead researcher on the SR26 work, Rohit and Peter, who led the SR27 work. And of course, I'd like to thank Brandy and Burkhard from John Innes Center and Sainsbury Laboratory. So they were the sort of the real pioneers of mut-RenSeq in wheat and were the brains behind a lot of these techniques. And also, I'd like to thank GRDC for funding most of this research, as well as Arbor Biosciences for hosting me today, and also processing our RenSeq samples. So if there's any questions, I'll leave it open to you guys.
Hello. Okay, great. Thank you so much, Tim. So we do have a little bit more time for some questions. So let me just pull up the few that popped in. If you have any more, please do continue to use that questions portal. Dev Padel asks about the assembly process that was used. How were the Illumina reads originally assembled? Was it a de novo assembly?
Yeah, straight de novo assembly. So originally we used the Macerca assembler, but we also found that the CLC Genomics Workbench assembler seemed to give us a lot better assemblies. Yeah.
Thanks. Sunish Segal asks about, I think it was the point that you were making at the end there about the bait capture set, which is currently the one you've been using is for NLRs. They're asking, is there a bait set for non-NLRs that you have or have been using?
No. So I think there are some people working on that for these TPK class of R gene. But yeah, I haven't been involved with that. Yeah.
Great. Arbor Biosciences does have a wheat exome panel that may or may not be relevant for that, or of course we could do a custom panel for whatever you know, genes are relevant. So definitely something that could be done if the sequences are available. Another question here from Douglas Blair, who asks, are the conserved introns that you used for finding R genes conserved across all R genes or just the species that you were using?
I wasn't too involved in the bait library design, but I assume it would just be in not just in wheat, but in the Triticeae clade. Yeah, because you are working at the level of DNA, so yeah, I don't imagine that they're conserved across all plant species. Yeah.
I have a just quick follow-up on that question. You mentioned at the beginning that having some intron content in the Triticea V3 bait set was a help to the downstream analysis. Can you just comment on what it was about having those introns present in the bait library that helped?
Well, I think they would help to recover like more contiguous, like a full gene, because yeah, as you saw with SR27 and PM1, it was actually fragmented within the intron. So those contigs weren't joined together because of that intron. There wasn't any bait representation. So I mean, with my work, we only used version 1 or version 2. So version 3, which was much more recent, we haven't had the opportunity to use that yet. But you know, that's the reason why the additional intron sequences were included in that library to overcome that problem, I imagine.
Yeah, makes sense for sure.
Yeah.
Question about the sequence depth that you're typically aiming for. Is there a minimum that you're looking for to confidently reconstruct an NLR sequence in your data sets?
A minimum what, sorry?
Read depth.
Oh, read depth. Yeah, typically, I mean, we aim for about, I mean, in our data sets, we usually got, you know, around 100x coverage on average. But, you know, I think at a bare minimum, you want at least 20x coverage. I mean, some, depending on the NLR, some sort of, you know, recover more read depth than others. But because you are calling Snips, you know, you want sufficient read coverage to be confident.
Yeah.
I'd say at least 20 to 50.
Great. I think I hit all of the questions that came in, so I just have one more, I believe, if there's no more. I'm curious about, it seems that you had a lot of great points about how the assembly quality needs to be, or the assemblies need to be quality controlled to make sure that you don't have these confounding factors of misalignment or deletion mutants,
Yeah.
mixing things up. So I'm curious, is it with the pipelines like Mutant Hunter and Nutrigo, are those cleaning it up to the point that you don't need to do additional curation, or are you typically like looking at your assemblies and curating them in some other way, like with visualization?
Yeah, so those pipelines, they don't clean up the assembly, the de novo assembly itself. They just sort of filter out any sort of noisy alignments from your search space. But you know, it is possible that if you have a misassembled sequence, that your target NLR could be in there. So that is one risk. So you do want to make sure you have a really robust, accurate assembly, and that comes down to, I guess, you know, the quality of your data, your sequencing data, and the assembler you use. So, but typically with these experiments, we didn't do too much like polishing on the de novo assembly. I think the read depth was enough that we got pretty sufficient contigs, and we found our target genes. So, yeah.
Great, thank you. One last question, and then we'll wrap things up. Sophia Sorazano is asking about, do you have any comments on the costs of the overall process and the time what it would take for a non-mono organism species? Just wondering if you have any thoughts on that.
Yes, I'm not. I think you guys would probably give a better idea of the costs for a RenSeq experiment. But definitely, it's you know, it saves a lot more time and costs than, you know, extensive mapping, using a mapping population and back libraries and all that. But yeah, if you're venturing into a new sort of non-model organism, I think you'd have to develop a bait library. So you'd need, you know, sufficient diversity panel to source those sequences from first.
Yeah, for sure. Yeah, either the same species or at least relatives, just to start with.
Yeah, that's right.
Yeah, that's a great question. You know, our team would be happy to dig deeper into that. So if you're please feel free to reach out to us directly, Sophia. I think that's it for the questions. So I will just one more. So yeah, absolutely. Thank you so much, Tim and Robert, for you know, being willing to participate in this originally workshop, now webinar. That was really excellent. It was really wonderful hearing from you both those details on those experiments. Thank you again to all the audience members for being with us today and submitting those great questions. As I mentioned before, we will be aiming to post this recording online as a future date, but in the meantime, if you have any questions for either Robert or Tim, they have agreed to share their email addresses here if you'd like to reach out to them directly about any questions about their research. And in addition, we have our Arbor Biosciences contact info there. Of course, visit our website, reach out to our team if any questions. And please do tune in again in March for part 2 of this series. There'll be 2 more presentations from researchers working in the RenSeq field. So we'll have Sanu Aurora from John Innes Center, as well as Ingo Hein from James Hutton Institute. So yeah, thank you again so much to our speakers for being with us today, everybody as well. And we wish everybody a pleasant rest of your day.
Thank you.
Thank you.
Thank you.